Today we are going to talk about next generation sequencing technologies. Before we talk about this revolutionary technology platform, let me share a story with you. So, when I was a grad student like you, I was finishing my masters in year around 2000, 2001. That time the various genome sequence projects were getting accomplished. It was big news that time, big breakthrough that time, that the human genome project, the first draft was completed, and many other model organisms and their sequences were also getting completed at the same time. Just imagine that first time we were getting information that what makes it human, what our genetic composition is about. It was really big science breakthrough, because first time we now know that how many genes are actually available to us, which makes it a human, which makes it so unique and different than other model organisms. Before that, there were only speculations that what makes human and how many genes we may have, but the first time the evidence for genes and our genetic composition, the genome, was first time revealed, right. It was definitely big revolution in the science field in general, and definitely in the biology and medicine area. But it's not end, right. It is not finished that time. So many times a technology shows us a promise and then it fades away. But what has happened in this area of genomics, that 2002 and 3 first time the draft human genome were released. But since then there has been tremendous advancement which has happened in this field of genomics, reaching to where we are, which we say as a next generation sequencing technology. This series of advancement happened for the first generation, second generation, third generation of next generation sequencing technology, and I must admit that advancement made by engineers and technologists, big data scientists and biologists together has really brought where we are currently in our magnitude of doing sequencing based experiments. So, what has changed from 2002 in 2018? Main thing which has changed is our speed, the speed at which we can now sequence the genome. Imagine the human genome project took more than 10 years time to accomplish. Now, within 2 days time or 48 hours time, you can do sequencing of a given individual with the next generation sequencing technology. That is humongous change. Additionally, what took $1 billion to accomplish one human genome sequence, now it takes maybe only $1,000 to accomplish the same sequencing technology based work. So, both cost wise as well as the speed wise as well as the accuracy wise, the next generation sequencing moving from the Sanger sequencing based platform have made tremendous revolution. So, I am sure now you are excited to know that what NGS technologies are, which are the currently big industry player, big industry technologies which are leading this field. And in this light, we have invited a guest, Dr. Atima Agarwal from Thermo Fisher Scientists, who is manager, a commercial service training and tech support at Thermo Fisher Scientific, who will be taking this lecture, and she will try to provide you the basics of next generation sequencing first, and then give you little more detail and specifics for one of the technologies of ion torrent. So, let me welcome Dr. Atima Agarwal for her lecture on NGS based technologies and its applications.
So, I am Atima Agarwal from Thermo Fisher. I have been with this company since many years now. So, if you remember or if you have heard, in pre-1990 era, the sequencing used to be done on gels with radioactive labels. Yeah. So, and it used to be a very cumbersome affair, but then it had contributed a lot to the science in those terms that first human genome was being sequenced on these systems, and there were many platforms, many of these capillary electrophoresis platforms. If you look into the pictures of Sanger Institute during that time, in lines there were these kind of systems, and they were generating sequences day and night, and that is how the first human genome got sequenced and got assembled. So, those were a lot of, like, I would say that benchmarks in sequencing. Suddenly somewhere during 2004, 2005, we started hearing about next generation sequencing. So if I were to talk about next generation sequencing, it is more aptly called as massively parallel sequencing. So with Sanger, what you're doing is that either you are running it on 96 well plates or you are running it on strip tubes. So one well is giving you one sequence. Yeah. So one sequence which is a stretch of 80 GC, and this can be as long as 1000 base pair, 1100 base pairs, and it can be as short as 100 base pair fragment. So one well gives you one sequence. Now suddenly with massively parallel sequencing, you can imagine what is happening is you have millions of wells and you have this sequencing per se happening in all those millions of wells, and that is how you are generating a lot of data per run. So that is massively parallel sequencing. So just to tell you that just because we are nowadays talking a lot about NGS, it is not that capillary sequencing has lost its charm or has lost its utility. It is still a very user-friendly technique for a lot of applications. So for things wherein you do not really require a lot of data, things can be done simpler. For a lot of microbial research or a lot of single gene based tests and all those things, they can be still very well and very efficiently being done on capillary sequences. Till date, this has been the gold standard technology for doing a lot of things like targeted. So you have certain amplicons to be sequenced, or you have a de novo organism to be sequenced to fill up those kind of assemblies. Then you have there are multiple fragment analysis applications wherein you are not actually sequencing the fragment, you are just looking into the size, and based on the size polymorphisms you are giving certain scientific answers. So this is about capillary sequencer. Coming to next generation sequencing, so now as I mentioned that this is a technology wherein you have millions of wells. So if you see that these are the chips which are used with ion technology, and you have millions of wells on these chips. So millions of wells, so you can imagine that each well has a sequence, and that is how you are generating millions of sequences in a run. Yeah. So the agenda, how we have planned out the session for you is that we are going to talk about our new series of NGS platforms, which is Ion Gene Studio S5 systems, the technology behind the Gene Studio S5s, and some of the applications. So this is the fastest sequencer which is available in terms of an NGS, which can produce, starting from once you have started your sequencing run, it can produce data in as little as 3.5 hours from a sequencing run. There are lots of different kinds of applications people want to work on NGS, and a lot of these samples are very challenging samples, like when you extract DNA or RNA from FFPE blocks, when you are working with liquid biopsies, like you really are looking at cell-free DNA, yeah, or you have very little RNA from a cell and still you want to study the whole transcriptome of this cell. So our technology is such that it is compatible with as low as 1 nanogram of DNA or RNA, and the backbone of this technology is AmpliSeq technology. We'll come to those details later. So coming to the workflow now, so how we are generating these millions of reads. So you start with nucleic acid. Now this nucleic acid can be DNA, can be RNA. So where do you think you would be starting with the DNA? What kind of experiments do you think you would be starting with the DNA on NGS? DNA as a sample. Whole genome sequencing, yes. Plasmids, yes. Targeting resequencing. So when you are not, so targeted resequencing is basically when you do not want to sequence the whole genomes, you just want to sequence some targeted regions of the genome, that is targeted resequencing, yeah. So where do you think you would be using RNA as your starting material? Transcriptomes, yeah. Again, like you're looking at some targeted transcripts, or you're looking at some targeted fusion events. Yeah. Anything else?
RNA-seq with protein atlas.
Yeah, RNA-seq. And RNA-seq, typically, why would you do an RNA-seq? You want to sequence.
More information that DNA will be hide. That information provides RNA.
Yeah, so this DNA is transcribed into RNA, and then hence there are more probabilities that this particular part of the genome is actually getting transcribed or further translated into a protein. But that's just a possibility. There are many non-coding RNAs also. Yeah, it's like small RNAs. Yeah, yeah, which are which are there involved in lot of regulation activities of how the transcriptome is shaping within a cell. Yeah. So people do a lot of different kinds of experiments. Now for what you would look out for in your NGS is that all these experiments, because the data requirement for all these experiments is very different. Yeah. So when you are doing a genome, it's a very straightforward calculation that you generally tend to generate 100x data. So whatever is the genome size, you multiply by 100. There are definitely ifs and buts around it. We'll not go into those details. But then the data requirements for these different kinds of experiments are very different. Yeah. So that is where you need that flexibility, that single NGS system can cater to all those requirements. So you started with your DNA or RNA, yeah, to begin with. So you isolated DNA, RNA from cells or from some culture isolates or whatever. So now once you've isolated this DNA and RNA, the next step is that you prepare a library. Now library is basically a collection of fragments, whether it is coming from DNA or coming from RNA, which you finally want to sequence. Yeah. So this can be an enriched portion. Yeah. So for targeted sequencing, you would need to enrich those targets first, and then only you can go for the sequencing. For whole genome sequencing, since your aim is to sequence whole genomes, so you do not need any enrichment. You will just extract DNA and you will go forward for library preparation. So library preparation is basically collection of fragments, whether DNA or RNA, which you finally want to sequence. Yeah. So what is the basic aim in library preparation is that you are now these next generation sequences, we talked about that you are generating 200 base pair reads, you are generating 400 base pair reads, you are generating 600 base pair reads. So supposingly we take an example of whole genome sequencing. Now what you are going to do is, with whole genome sequencing, your aim is to sequence that genome as fast as possible. Yeah. And with as bigger reads as possible. So probably you will pick up a 400 or a 600 base pair chemistry, and so what you need to do with the genome is you shear this genome. Yeah, you break this genome into smaller pieces. So that can be done by enzymatic treatment, or that can be done by sonication. So once you've broken down this genome, now these are the fragments which you finally want to sequence, but you don't know what is there, so you need to have some adapters, yeah, or linkers, which will help you facilitate sequencing these regions. Yeah. So in library preparation, what you're doing is you are enriching the region of interest, and second is you are ligating it at both the ends. You are ligating it with linkers. These linkers are double-stranded DNA fragments, which are basically exploited so that the primer can then sit on these kind of fragments, and then so you have your fragment of interest in between, you link the adapters at both the ends, and so why? So that these adapter sequences you know, because these are coming from a chemistry and we tell you that these are the adapter sequences, and then the primer comes and sits on these adapters so that you can sequence this region in between. Yeah. So that is basically, so with library preparation what you are doing is you are enriching your region of interest and you are ligating these fragments to adapters. Yeah. Then comes template preparation. So basically now these are the things which are specific to a technology. So in template preparation what you are doing is that you are taking these library molecules and you are amplifying onto ion spheres. So, we are talking about ion torrent technology. So, we are using ion spheres as the mode. These ion spheres get coated onto those or get loaded onto those chips, and finally these ion spheres are carrying those regions of interest or those library molecules from which you are generating all those reads. Yeah? So, that process of taking these library molecules and amplifying them onto the ion sphere is called template preparation. Then comes sequencing. Now, you have your ion spheres which are templated, which have library molecules amplified onto them, and now you want to sequence. So, you load these ion spheres onto the chip, and then you generate the data. So, once you have loaded, you initialize your sequencer, which we discussed is going to take only 15 minutes, and then your sequencing run goes for anywhere from 2.5 to 4.5 hours depending on the kind of read length you are targeting. So, talking about the instrumentation which is required in this workflow. So, we have, so library preparation can be done manually, can be done on Iron Chef. There are some parts of library which can be done on Iron Chef. Yeah. So, this is an automated way of preparing libraries. Otherwise, you have manual way of preparing libraries, wherein you are using certain magnetic beads, certain library preparation kits from us, and the process generally for targeted sequencing or for whole genome sequencing, the process is around 4 to 4.5 hours for manual, and probably for RNA-seq it's a day's protocol, because you have to do certain levels of enrichment, and you and you have to do more QCs in that. So, if you talk about manual preparing libraries manually, so that's the kind of time frame. Otherwise, you can use Iron Chef for preparing your targeted sequencing libraries automatically. Then comes the template preparation. Now, as we discussed, during template preparation, you are loading these library molecules onto the ion spheres, and then finally these ion spheres are loaded onto the chip. Now, these loaded chips you take from here and you just load it in on this system, and they are good to go for sequencing. So, just coming into a few details on library preparation. Now, how we go about library preparation is, since you are targeting variations at the DNA level as well as at the RNA level, you isolate DNA and RNA. Yeah. Then you reverse transcribe this RNA. So, now you have DNA or cDNA. So, you take this primer pool and you use this DNA and cDNA as your target. All these genes, yeah, get amplified at one go, and then since the primers are coming from conserved regions wherein you do not have any hotspots or any kind of variations to be looked into, we partially digest these primer sequences and then we ligate adapters. Now, when we say that you can do smaller experiments also, so you have a 52 gene panel and you have some 12 samples to be done. So, what you are going to do is, you index these 12 samples by 12 different barcodes. Now, these barcodes, so when we said that library preparation is all about enrichment of the targets and ligating adapters. So, when we are ligating these adapters, you are using barcoded adapters to differentiate one sample from another. Yeah. Now, this barcode is a 10 base pair unique sequence which is present in the adapter. Yeah, which will finally help you to differentiate between different kinds of samples which you are loading on one chip. So, once you have ligated these adapters, you pool all these libraries. Now, you are working with probably 6 samples on a chip. So, based on the number of samples you plan to multiplex on a chip, you are going to use the number of barcodes. Yeah. Once you have done that, now you want equal amount of data for all those libraries. What you are going to do is, in terms of their molarity concentration, you will use equal molar concentration for all those libraries and make one pool of that. But the aim is that when you are doing small RNA sequencing for all these samples, you are aiming that you generate equal amount of data for all these samples, but because otherwise probably some of the samples have worked out pretty well and some will not. So to ensure that, you need to pool all these libraries in equimolar proportion. So once you've pooled that, after your library preparation step, now you started with 12 samples or you started with 20 samples, now that becomes one sample. Yeah. And now how are you going to differentiate? Now because it is now one sample, but you started with 20 samples. So when you generate the data, how are you going to differentiate? Yes, we used barcoded adapters. So the system is first thing it's going to use when it sequences the barcode sequences, and then it sequences your region of interest. And then it makes the bins that, okay, this read belongs to this sample and it is in this bin. Once we are through with our library preparation, then the next step is template preparation. So in template preparation, the aim is that you clonally amplify this. So you have prepared, you started with targeted sequencing, or you started with whole genome sequencing, or you started with an RNA-seq experiment. You have tons of millions of target molecules which are to be sequenced. So what we are aiming during template preparation is that one library molecule gets clonally amplified onto one ion sphere. So this is the basic aim of this particular step, and so this library molecule gets amplified onto this ion sphere, and this is one clone which is getting amplified all over on the ion sphere. Then there could be some ion spheres which have not got amplified. Yeah? So you do not want to load them on the chip, so you negate all those ion spheres at the enrichment step. Yeah? So what is happening is that we are using ion spheres which have an oligo which is linked to these. So this oligo is complementary to one of the adapters which you used during library preparation. So if we go back a few slides, so we used two adapters, P1 and one was barcoded adapter. So, and we mentioned earlier that we are basically exploiting these adapter sequences for us to enable the sequence to sequence these millions of fragments later on one chip. So we are using this particular P1 adapter sequence, yeah, for the library molecules to complementary go and bind onto the ion sphere. So now if we go back to that slide, so these ion spheres are loaded or are coated with oligos which are complementary to the P1 adapter. So when these emulsions are being created, you are basically making these water molecules which are surrounded by oil, and you are trying to amplify these library molecules. So when this emulsion is being created, this is a random distribution of these ion spheres with the library molecule and enough PCR components for it to amplify this library molecule onto this ion sphere. So what is happening is, a library molecule is coming, it is getting denatured and it is sitting on that complementary sequence which is complementary to the P1 adapter, and then it goes ahead and it sequences this part, and that's how you sequence. Now this goes through a cycle of denaturation, annealing and extension like your normal PCR, but while we are talking, all this has been, so you are not manually involved with all this process, all this is being done by these systems, either semi-automatedly or fully automatedly. So then when it gets denatured, it then again binds to another oligo on that same bead, and you then, that's how you are generating, you are clonally amplifying this particular molecule onto the ion sphere. So what is, if we go into the details, what is happening is you have an emulsion oil, then you have a library molecule, you have some primers, this is your ion sphere, this library molecule gets denatured, it gets bound to the ion sphere, it gets extended, again gets denatured. Now this template goes and binds to another oligo, and finally you have this one library molecule clonally amplified onto this ion sphere. Yeah. So now we said that there could be some molecules or some of those water droplets, when the emulsion was being created, they did not get a library molecule. So they will not get amplified also, but we do not want to take them on the chip because they are still going to occupy some space on the chip. So why to lose on that data? So what we are doing at the enrichment step is, when we were doing amplification on the ion spheres, one of the primer was biotinylated. So now all the amplified ion spheres are biotinylated. We are using streptobidin coated beads to fish all the amplified molecules, and rest all molecules remain in the solution. Yeah. And then we just go through. So this is primarily your ion sphere. This will have these molecules all over because we clonally amplified. So you use streptobidin coated beads, and this streptobidin coated beads are magnetic. So with the help of a magnet, all these are fished out, and then you have only the beads which have amplified, which are finally to be loaded on the chip. So this is the enrichment part of the template preparation. Then what is happening at the sequencing level? Now I showed you that chip. That chip has millions of wells, and each well is generating a sequence. So this is your chip. It has millions of wells, and if we are now to consider that this is one well, this one well has one ion sphere which is loaded with one kind of template. So what is happening during sequencing is that when ion chef is preparing the sample for the sequencing, it is already adding the primer and polymerase to the chip itself. Now what else it needs for extension? DNTPs, yeah. So what the system does is, it is adding DNTPs sequentially onto the chip, and then naturally we know that whenever a DNTP gets incorporated or gets polymerized into a growing chain of DNA, there is a bond formed between the phosphate group and the hydroxyl group, yeah, and this phosphodiester bond, when formed, this releases a hydrogen ion, yeah. So now we coated these library molecules onto this ion sphere. So this same event, so supposingly the first base here is an A, yeah. So you had flown DTTP, so it will have a complement, it is a complementary nucleotide, it will bind and it will release hydrogen ions, and these hydrogen ions get converted into a voltage signal which is finally being captured. So this ion basically is these release of so many hydrogen ions in one well are bringing up a change in pH for that particular well, yeah. And when the pH is being changed for that particular well, that change in pH is being recorded as a change in voltage. So basically you are converting a chemical information into a digital information, and that is how you are saying that a signal has been received and a base has been called. Now supposingly, now we said that T is your, or A is your first base, and the system had flown a DGTP. So now that is, G is not going to bind to A, yeah. So what will happen is, there will be no binding, no hydrogen ion release, so no change in pH, no signal detected by the system. So we will just see this by a cartoon. So considering that this is your one ion sphere, these are your adapters, yeah. This is the barcoded adapter, and this is your adapter which you used for ligating these sequences onto the ion sphere. Then what is happening is, your primer is coming and binding here, you add the DNTP sequentially. Now what will happen is, this will bind, you will have a change in pH which will be recorded by the system, and the system knows that it is flowing a DTTP at this particular time, and that is why it can associate this signal with an A, yeah. So we are not using any fluorescently labeled DNTPs, so this is a simple chemistry. Now what will happen in the next, so we had flown in an A, and the next target base is G. They are not complementary, so it will not bind, it will get washed off. Then again, so likewise you will build up the sequence. Now what will happen at this step? Supposingly we are flowing a DTTP, what will happen? Sorry? Yeah, since there are two A's in a stretch and there is nothing stopping it, it is a growing chain, yeah, so you will have double the signal. Yeah. So there are double molecules, double number of hydrogen ion release, which will bring up a double change in the voltage, and that is how it is being recorded as signal, which is double, and that is why you can tell that there were two consecutive A's in the sequence. So then this, by adding these DNTPs sequentially, finally the system is creating a sequence which is finally being read as an ionogram. So this ionogram is again being read by the sequencer. This has to be read from left to right and for the number of bases. So whether you get a signal or not, that will define the sequence of the DNTPs. The intensity of the signal will define how many of those DNTP or how many of those A's or T's were in a row.
So, hope you got a glimpse of next generation sequencing technologies which has really revolutionized the field of genomics. You were given the basics and some possible applications using this technology platform, and one specific technology of ion torrent was also illustrated and discussed. These concepts will be again covered in more detail in the following lectures, when we have invited some more industry experts representing different technology platforms, who will also provide you the basics and the current status of these technologies. So, see you in the next lecture. Thank you.